Hello and welcome to another very entertaining video. I'd like to give just a brief introduction, but show a little bit more detail than we have in the past about the renin angiotensin aldosterone system. And if you recall from previous discussions, this is all about regulating blood pressure. Just consider for a second the last time you felt dizzy or you were lightheaded. Maybe you actually fainted. It could be blurred vision, nausea, fatigue, lack of concentration. All of these are associated with low blood pressure. On the other side, high blood pressure, at least chronically, can lead to kidney damage and heart problems, heart disease. It's often caused by vascular disease, and this is going to lead to all sorts of downstream complications. And so homeostatic balance of blood pressure is critical not only to everyday life and all the activities we do, but also to longevity. And at the heart of long-term regulation is the renin angiotensin aldosterone system. Not surprisingly, this system is initiated in the kidney. This makes sense to us because the kidney is probably one of the organs most prone to blood pressure. In our previous discussion on filtration at the glomerulus, pressure is one of the key attributes that drives your overall rate of GFR. Too much pressure, and these things can be damaged. Too little pressure, and we're not getting enough filtration. We're not cleaning the blood as we should. And so having the kidney at the core of blood pressure regulation makes sense. Now, at the heart of this discussion, we have to talk about a cell population called juxtaglomerular cells. As the name implies, these cells are hanging out near the glomerulus and are intimately involved in regulating specifically the afferent arteriole feeding into the glomerulus in order to regulate the glomerular hydrostatic pressure and therefore regulate GFR. And it's from these cells that we can get the release of a hormone called renin. Now, to paint the big picture, what I want you to understand is renin's overall job, once released from the juxtaglomerular cells, is to increase blood pressure. So let's think about the signals that the juxtaglomerular cells could receive that could induce renin release. The first trigger is simply low blood pressure. This is sensed by stretch receptors within the juxtaglomerular cells themselves in a manner analogous to baroreceptors that we've talked about. The difference being baroreceptors are sensing blood pressure for the entire body. These stretch receptors are just sensing pressure into these afferent arterioles within the kidney. A second trigger could be signals from the macula densa. And if you recall from a previous video, the macula densa is a specialized cluster of cells at the end of the loop of Henle as we transition into the distal convoluted tubule, whose job it is to sense more or less sodium concentrations coming out of the loop of Henle and use those sodium concentrations to interpolate blood pressure. And generally speaking, we like to think that decreased salt concentrations correlates with low blood pressure. And so if the macula densa is detecting decreased salt concentrations, it will signal through paracrine signaling to the juxtaglomerular cells, causing them to release renin. Now there's one more trigger in day-to-day life that we need to be aware of. This is simply sympathetic tone. Whether I'm exercising, for 45 minutes on a treadmill, whatever it is, in that moment, I need increased blood pressure. And so sympathetic tone associated with fight or flight or exercise or any of those characteristic sympathetic drivers will lead to an increase in renin. Because remember, all of these things, what do they want to do? They're trying to increase blood pressure. All right, now that we have that in mind, let's focus on renin itself to help understand what it's doing. To do that, we're going to go away from the kidney for a second, and we're going to go into the liver. The liver, of course, is the source of the vast majority of the proteins that are circulating in my blood. And one of those proteins, made in the liver as a zymogen, is a protein called angiotensinogen. We've talked about zymogens before. These are simply proteins that are made in an inactive state, and they require activation from another enzyme before they themselves become active. And you guessed it, that's what renin's job is. So angiotensin released from the liver, circulating through the blood at all times, but in the presence of renin, angiotensin is converted, or angiotensinogen is converted to angiotensin 1. And the role that renin plays in this simply is a big pair of scissors, where renin cleaves off a portion of the original angiotensinogen zymogen, and by cleaving off that portion, converts it to angiotensin 1. Now, while angiotensin 1 has some biologic activity, for the most part, it's not nearly as efficient at what we want it to do compared to angiotensin 2. Angiotensin 1 is converted to angiotensin 2 by another cleavage event, and this time the pair of scissors is an enzyme called angiotensin converting enzyme, often referred to as ACE. Most ACE is localized in the endothelial cells of the lungs, and this is a common enzyme target for therapeutics whose goal it is to lower blood pressure. In other words, if I can target and block ACE, then I can prevent the conversion of angiotensin 1 to angiotensin 2, and if I lower the amounts of angiotensin 2, I'm going to lower blood pressure. Because remember, the whole purpose of the renin angiotensin aldosterone system is to increase blood pressure. All right, so what's so magic about angiotensin 2? What does it do to increase blood pressure? There really are three things that we want to focus on. The first target of angiotensin 2, smooth muscle surrounding the small arteries and arterioles. And here, angiotensin 2 causes vasoconstriction. We can see this essentially by a squeezing of these small arteries and arterioles. And we've talked previously how a decrease in radius leads to a very significant increase in resistance. And that's how blood pressure will increase. The second two targets of angiotensin 2 that we want to talk about are both hormones. The first of these targets is the hypothalamus, which will, from the posterior pituitary, release a hormone called antidiuretic hormone, abbreviated ADH. And we'll talk in just a moment what ADH is going to do to help increase blood pressure. But in order to do that, we have to understand the third target as well. And that third target is the adrenal gland. This is a gland sitting right on top of the kidney. And what we're specifically interested in here is the release of aldosterone. And for the purpose of blood pressure regulation, what we're talking about here, let's now show how aldosterone and ADH work together to increase blood pressure. So both ADH and aldosterone share the same target. They target a cell in the collecting duct of the kidney called the principal cell. Now, as just a quick reminder, where are we? We can break the nephron of the kidney, the functional unit of the kidney, down into several different areas, starting with filtration in the glomerulus, where filtrate enters the Bowman's capsule. We then go through the proximal convoluted tubule, where most reabsorption takes place. Then the loop of Henle, where we specifically focus on water and salt reabsorption, pass through the macula densa, and then in the distal convoluted tubule and the collecting duct, where the filtrate is finally destined for excretion into the toilet. And the principal cells that we're talking about are hanging out in this region, the distal convoluted tubule and the collecting duct. So let's zoom in, where we can see the lumen of the collecting duct. This is where filtrate is passing through. And of course, this lumen really is shaped and lined by cells, and these are the principal cells. Now, they're fairly close to vasculature, so let's add a blood vessel up here. As endocrine hormones, ADH and aldosterone are traveling through the blood to get to the kidney. So this is where I'm going to find my ADH and aldosterone. Now, let's take a step back and let's make sure we set the stage. We're in the collecting duct. How is this going to regulate blood pressure? Well, remember, if I can increase blood volume, this will lead to increased blood pressure. And how do I increase blood volume? By increasing water reabsorption. And how do I move water? By moving salt. So, as we look at the function of aldosterone, for our purposes, aldosterone does two things. Let's zoom in and take a look at this. One of aldosterone's target is the sodium-potassium pump. And remember, the sodium-potassium pump is responsible for pumping 3 sodium out and 2 potassium in. It's ATP-driven primary active transport. As aldosterone targets this, it increases both the number and rate of these primary active transporters so that I'm pulling more sodium out of the cell. Of course, we're going to have channels specific for sodium, and opening these channels, increasing the number of them, is another role for aldosterone, so that sodium comes in down its concentration gradient as it's actively pumped from the filtrate back into the blood by the sodium-potassium pump. Now, it's true that potassium is going in the opposite direction, but there are more ions, more sodiums going into the blood than there are potassiums leaving. And so there's a net gain in the amount of salt moving from the filtrate into the blood. This increases the osmotic gradient. And remember, what does water want to do? Water wants to follow salt. It follows a solute concentration gradient. And so this increases the desire for water to move. This is where ADH comes in. Of course, we've been doing all this in the other principal cells as well, right? So we're opening up sodium channels. Sodium's coming in. Within these cells, we see exocytic vesicles loaded with aquaporins. Remember, aquaporins are leak channels essentially for water. Water can't go through the cell membrane, it's polar. But the aquaporin allows for facilitated diffusion of water, and the role of ADH is to cause exocytosis. ADH will bring these vesicles to the luminal surface and cause them to fuse with the luminal membrane. And what this does is it places aquaporins on the surface of these cells, the luminal surface. Now we can see how both of these are working together to increase the reuptake of water. Aldosterone creates a steeper salt gradient, while ADH increases water permeability so that water can follow the salt gradient created by aldosterone. As these two work together, water reabsorption is increased, putting more water in the blood. And remember, if I increase blood volume, I'm increasing blood pressure. Now, just as a side note, I want to mention that aldosterone can work independent of ADH as well. While both ADH and aldosterone are part of the renin angiotensin aldosterone system, I can also trigger aldosterone, and only aldosterone, with high potassium levels. In other words, increased potassium or hyperkalemia is also a trigger for aldosterone release. And I hope from this picture you can see that independent of ADH, in this way, aldosterone can move salt, but water won't follow because the cells are impermeable to water. And the net result, if we again focus on what's going on right here with the sodium-potassium pump, is that we're taking potassium out of the blood and putting it into the filtrate. Where does this potassium end up? The potassium goes through leak channels out into the filtrate. So this is the body's primary mechanism for regulating high potassium. However, with this mechanism now, I hope you can appreciate, if I'm inducing a chronic state of low blood pressure, for example, if I'm treating somebody with a loop diuretic, which is going to decrease water reabsorption as a means of treating high blood pressure, then the renin angiotensin aldosterone system is going to engage because it senses the low blood pressure. And if I run the aldosterone for too long, I pump all of my potassium out. So chronic low blood pressure can actually lead to a state of hypokalemia. And this is because the chronic activation and release of aldosterone would continuously push potassium out. This is why people who are on drugs like bumetanide, furosemide, these loop diuretics that are regulating blood pressure by causing decreased water reabsorption, with these drugs, you also have to be on potassium supplements because your potassium is being wasted through this system. All right, if we zoom out and look at the big picture, the kidney plays a critical role in blood pressure regulation, and its go-to mechanism is the release of renin from juxtaglomerular cells. The three principal triggers that cause this release are a measure of low blood pressure as detected by stretch receptors within the juxtaglomerular cells, signals from the macula densa, where decreased salt concentrations are interpolated as decreased blood pressure, and also sympathetic tone, where my specific job is to try and increase blood pressure. With the release of renin, renin does one thing. It acts as a pair of scissors to cut angiotensinogen, a zymogen released from the liver. By cleaving this, we turn it into angiotensin 1, which is still biologically relatively inert, but can then be converted into angiotensin 2 by ACE. Angiotensin 2 is my biologically active molecule, and the three targets that we talked about, number one, small arteries and arterioles, where it causes vasoconstriction, leading to increased resistance and therefore increased blood pressure. And then also the hypothalamus, where the ADH is released from the posterior pituitary, and the adrenal gland, causing aldosterone release. ADH and aldosterone work together to target principal cells in the kidney, where aldosterone drives an increased reuptake of sodium, thus increasing the osmotic gradient for water, and ADH increases water permeability by causing the exocytosis of aquaporins. Thus, the combined effects of an increased salt gradient and increased water permeability lead to an increased reuptake of water, and by increasing blood volume, I increase blood pressure.